Hi, everyone, and welcome. I'd like to start by introducing myself. My name is Belinda Griffin, and I'm the program manager for the Transcelerate Digital Data Flow Initiative. And I'm going to be serving as your moderator for today's inaugural DDF Solutions Showcase, which will feature NNIT and a joint presentation from EQTY and ClinLine. Just to give you a quick sense of the agenda today, we will very quickly go through some logistics and ground rules for today's session. And then we'll move right into the presentations with NNIT going first, followed by EQTY and ClinLine. We'll then follow that up with an open Q&A session with the panelists. If we could go to the next slide. So as I mentioned, first today we'll have NNIT presenting. We'll have Cor, Brittany, and Sam representing NNIT. And they'll be followed by Ali and Berber from, as I mentioned, EQTY and ClinLine. And as I mentioned, following the formal presentations from both groups, we'll have a question and answer session. So just a few logistics for today's session. All participants will be muted, but you may submit a question at any time during the webinar using the button that you see in the top right-hand corner that has the little question mark icon in it. If your question is directed toward a specific presenter, please denote that when submitting your question. We will endeavor to get through as many questions as possible, but please understand due to the volume of questions, we may not be able to get to all of them today. And then just a note on the ground rules for today's webinar. I'm not going to read through all of these. I would like to just highlight the last section around some of the question topics that we're not able to address today. So we would not be able to address any questions around specific vendors or other business partners with whom member companies are working. We can't answer any questions about costs of using or implementing Transcelerate assets or tools. And we can't answer any questions about which member companies are using or going to use any Transcelerate solutions or any commercial product or service. Finally, before we turn the floor over to NNIT, I'd just like to acknowledge our two sponsors for our webinar today. Our first sponsor is Transcelerate, which is a not-for-profit entity focused on fostering collaboration across the biopharmaceutical R&D community. And in this capacity, Transcelerate is the driving entity behind the Digital Data Flow Initiative. Our co-sponsor for today's webinar is CDISC. CDISC is a leading entity in the development of clinical research data standards, and as such is leading the development of the Unified Study Definition Model, or USDM standard, that is foundational to DDF. On that note, without further ado, I'd like to turn it over to our presenters from NNIT.
All right, so what we're going to talk about today is how FHIR and the USDM can unlock the full benefits of digitized protocols. So we'll start with a brief introduction before talking about what the USDM and protocol digitalization are and what they mean for the industry. Then we'll talk about how FHIR and or talk a little bit about FHIR, what it is, and how it can be used as a vehicle for moving data around the clinical trial ecosystem. Lastly, we'll dive into the practical solution of how mapping FHIR to the data from digital protocols using the USDM can be what we think is a game changer for interoperability. All right, so just a little background. My name is Brittany Walker. I'm a clinical advisory managing consultant with NNIT, and joining me today is Cor Hedel, who is a principal consultant on our R&D solutions team at NNIT, and also Sam Thompson, who is a regulatory advisory managing consultant here at NNIT. So Cor, I'll let you talk next through this slide.
Alrighty, thank you so much, and also thanks to Transcelerate for setting up this thing today. First of all, would like to maybe elaborate a bit on some of the overall grand strategic objectives of this use case we will be presenting today. Because I don't think I am offending anyone if I'm saying that life sciences today is indeed a fairly fragmented industry from a data perspective. So this is what this slide is trying to show, that typically we see that we have these dedicated systems that are generating insights, actions, or data in a silo. And thus, that will of course lead to a fragmented information space. Therefore, we at NNIT propose that we should all strive towards a future where it is interoperable data that is driving the show. So this is a slide showcasing that. We want to move towards a future where the data flows into a centralized database and generates a unified information flow, meaning that decision makers will have access to the full information. And with that introduction, I will again hand over the word to you, Brittany.
All right. So a bit more background about what the USDM is. The USDM provides a unified framework to define key elements of a clinical study, like the eligibility criteria, schedule of activities, and other essential components. And so it harmonizes various data elements used in clinical protocols by aligning them with some of the existing CDISC standards, like SDTM, or the Study Data Tabulation Model, or ADAM, the Analysis Data Model. So this can ensure consistency in how clinical data is represented and shared. So in simpler terms, the model is designed to facilitate seamless exchange of information between stakeholders, like sponsors, CROs, sites, and regulators, by providing a common language and structure for study definitions. So this standardization can enable better interoperability across the different systems involved in clinical trials. So what is protocol digitization? This refers to the process of converting traditional clinical trial protocols into digital formats like XML or JSON. This transformation is crucial for modernizing clinical trials and adopting full protocol digitalization, which means that not only is the protocol digitized, but digital solutions are also leveraged for protocol execution and management processes as well. So protocol information is needed by virtually every system used to plan, run, analyze, and report on a clinical trial. Still, it's often trapped in these documents that are not machine readable, and populating information in the downstream systems requires repeated human interpretation and manual data entry. So we and many others, hence the Digital Data Flow Initiative, think that the process of setting up these other systems can be done in a more efficient and automated manner in the future using the modern standards and tools that we have available. So then kind of how they fit together, the USDM and digitalization. So the Unified Study Definitions model plays a crucial role and provides some great benefits in enabling digitalization. To look at some of the more specific benefits, first, the USDM provides that standardized data model for defining clinical study protocols and ensures that all of the protocol information is captured in a consistent machine-readable format. Second, by using the USDM, different systems involved in clinical trials can seamlessly exchange protocol information. This reduces the need for that manual data entry and human interpretation down the line, which can be very error-prone and time-consuming. Third, the USDM supports that automation of various processes in clinical trials, like automatically setting up study designs, managing patient data collection schedules, and integrating with electronic data capture systems, or EDCs, using standard APIs. Fourth, digitizing protocols with the USDM improves efficiency by streamlining the setup and execution of clinical trials, which can lead to faster study startup times and reduced cycle times for data processing. And lastly, the structured data provided by the USDM enables advanced analytics like AI, machine learning, which can enhance decision-making and optimize trial outcomes. All right, so some of the challenges. There are some barriers to adoption of digitalization in the industry, and that kind of optimal digital data flow framework that the industry is grappling with. So first, there's a lack of standardization and interoperability. There are various formats that data is kept in these disparate clinical trial systems, and the formats may be incompatible. There's an inconsistent use of standards, especially in those older legacy systems. Second, there's a lack of support for major digital transformation, even as the R&D industry is modernizing. So the solution that we're proposing is sort of the future state where everything is standardized and connected, and this is certainly an expensive and daunting undertaking. In such a chaotic and fast-moving industry, there's often hesitation to make big changes in processes that may cause disruption. We're slowly chipping away at this hesitation by highlighting some of the benefits of the future state. Lastly, I think this is partially caused by the resistance of change, is the challenge of maintaining regulatory compliance while implementing new processes and standards. It's kind of tough to be among the first when the regulations haven't quite yet caught up, or we're unsure where the regulations will end up. And then there's also the concerns about data privacy and security in a world where data is freely flowing from one system to another. So the solution that we're proposing is a methodology that we think can streamline clinical protocol data exchange by mapping the USDM to Fast Healthcare Interoperability Resources, or FHIR. USDM alone provides that standard that can be used to reduce errors and inconsistency as the world moves towards digitized protocols, which will unlock the ability to automate many of the workflows and reduce that manual effort that I mentioned a few minutes ago. So by mapping USDM to FHIR standards, this solution ensures seamless integration with those critical downstream systems like your CTMS or your EDC, while significantly reducing the need for manual input. So this can enhance accuracy, traceability, and compliance, and provide that digital-first source of truth for clinical protocol data. So this approach will enable advanced analytics on that unified data, improving data integrity and supporting that informed decision-making throughout the clinical trial lifecycle. So let's look a bit more closely about how this would work in practice by reviewing some of the information from a protocol and where it needs to go. So the clinical protocol serves as the source of truth for pretty much all of the operational systems that support a clinical trial. The visit schedules and procedures go into the CTMS. The list of lab tests and assays required by the protocol go into the laboratory information system, and the randomization schema and treatment arms may go into the IRT system. Today, the process for getting all of this information is done either by manual input by users for each of these systems, or through custom integrations that can be difficult to set up and maintain. So we're proposing that FHIR can be that vehicle for moving a lot of this information to the different systems that it needs to get to in order to support the protocol setup and execution. The use case that we're going to focus on a little bit more is on the flow of information from the protocol to the EDC. So an EDC, electronic data capture system, is a software application used in clinical trials to collect, manage, and store clinical trial data about the subjects in a digital format. So the EDC system uses the protocol to create CRFs defining which data points must be collected at each study visit. So each study needs a unique EDC setup to properly capture the data that needs to be collected during the trial. Today, the configuration and setup process for an EDC typically involves collaboration among clinical operations teams, data managers, biostatisticians, and technical professionals, and the process can be cumbersome, lengthy, and error-prone. So we're going to talk more about how FHIR can be used as that vehicle for moving the information from the protocol to support the EDC setup. So I'll hand it over to Core here to talk more about FHIR and how it can be used.
Yeah, thank you so much. So first, just a brief introduction to what FHIR is. FHIR stands for Fast Healthcare Interoperability Resources, and as the name suggests, it originated in healthcare. But we at NNIT see a bigger and bigger push towards adopting FHIR in life sciences, of course, mainly in the clinical sphere, but certainly also in the regulatory sphere with the increased proliferation of FHIR-based data standards, such as the FDA-sponsored PQCMC initiatives, the pharmaceutical quality standard, SPL, CDISC, and others. So what is FHIR then? It is essentially a universal language that enables systems to talk to each other. So for example, if I switch to my mother tongue, Danish, så tror jeg vil have svært ved at forstå, hvad jeg sagde, meaning you would have a hard time understanding what I was saying. And it is the same with systems. So we need this universal language to basically tie everything together. Like Brittany just showed before, we have our protocol template in the middle. It needs to pass out this information to its auxiliary downstream systems. I like to think about it as a general sending orders down to its soldiers so they know what to do. So in essence, FHIR enables streamlined data exchange across various systems like the EDCs, CTMSs, and EHRs, resulting in much more efficient workflows and quicker insights. And if you recall the introduction slide I presented, this is essentially a way of moving towards that interoperable future where we simply say no to these manual or semi-manual processes, document-based workflows, and what have you. So with FHIR, we are currently at the R5, that's the newest version. It contains 157 resources, and a resource is essentially a collection of data points that allows us to describe a context in an interoperable manner. So an example could be the medicinal product definition FHIR resource, which contains information about medicinal products, of course. We also have the R4, which is the normative FHIR version. It is widely adopted specifically in the US healthcare system. So in essence, the modular structure of FHIR allows very diverse data types and systems for that matter to be standardized, supporting interoperability for a wide range of use cases. It is also built on modern web technologies such as RESTful APIs, JSON, and XMLs, meaning that a developer who may or may not know much about life sciences or healthcare can actually implement FHIR fairly quickly. So I guess the big picture here is that FHIR is a strategic enabler in breaking down data silos to accelerate data-driven decision-making and thus ensure that global data consistency is ensured. So this is our proposal for how to achieve this. In clinical trials, it is indeed very crucial to ensure accurate and timely data transfer. So this is about moving the right protocol information to the right systems efficiently. So the goal here is essentially to convert specific digital protocol elements into actionable data for each downstream system without overhead. So in this flow, the digital protocol concept acts as the single source of truth for capturing essential protocol elements. And again, FHIR acts as the standard format to make sure the protocol data is easily handled for downstream integration. And then, this is a Python function. It could be a bunch of other things, but wanted to keep this fairly agnostic. But rest assured, there are dedicated systems that can achieve just the same. But essentially, the Python function here is a sort of intelligence or mapping layer. It basically validates and transforms protocol elements into a format that the downstream system can consume. And then we have the cloud storage. It could be something similar to Amazon S3 or something similar to that. It serves as a checkpoint, meaning it will store data that failed validation. It can flag any errors in the ingestion. And of course, it can also hold a copy of the data for audit trail / GXP purposes. So essentially, this flow minimizes the manual data handling and reduces errors related here too. So it makes sure that any EDC system can quickly receive the protocol data that they need in the correct format. And just building on top of that, we know that clinical trials like Brittany just showed us involve multiple systems, with each requiring their own unique protocol elements. So here the architecture expands the flow to distribute the data not just to the EDC, but also to CTMS, EHRs, and LIMS. So if we look at where we are today, a architecture such as this one would be needed, but I wouldn't be surprised if we see a push actually from the legislature. We saw in healthcare back in 2022 that it got mandated that all EHR systems in healthcare must have a FHIR connector or FHIR API. And I think maybe the same will happen within life sciences at some point. And my point is basically that once that happens, the seamless exchange of information will become much, much easier. But in order to start, we need to basically do bespoke integrations to each system. So while not ideal, it is indeed a start. So again, here each target system receives the relevant protocol data it needs. Again, the general passing down orders to its infantry soldiers. It could be patient info to the EHR and LIMS for lab samples. So again, by routing the correct data to the right system, this architecture enhances data quality and consistency. And this efficiency ensures that all systems align with the protocol's requirements, accelerating clinical trial processes from recruitment to lab testing. So ultimately, this model empowers clinical trial teams to focus on what truly matters, delivering life-saving treatments faster and more efficiently to patients. And with that, I would like to hand over the word to Sam Thompson, who has been working day and night on building some of these mapping schemas. Take it away, Sam.
Hi, everybody. Thank you so much for joining, and thank you, Core. Yeah, I'm going to start off by taking a quick look at the implementation guide for HR FHIR. This shows the most current complete version of HL7 FHIR, which is R4, release 4. And we can see a list of resources which contain a ton of information. And so the challenge that we took on was mapping all of the information from the USDM implementation guide into FHIR. And as you probably know, there's a a lot of information in the USDM model. Fire has well over 2,000 data points, and that's spread out across 145 resources. And it covers, as Core mentioned, it covers everything from healthcare to regulatory, labeling, clinical, PQ. So there's an awful lot of information to try and manage one-to-one. So, as a vegetarian, I always ask, how do I eat a 2,000-pound block of tofu? And that's one bite at a time. So as Brittany mentioned and showed sort of an example of sending information to an EDC. So let's start with some of the population information. We can look at, again, from the implementation guide, and if you have a sharp eye, you'll see that when we built these slides, that was when the 3.0 IG was in place. So USDM now has implementation guide version 4.0. This is still a valid diagram. So still, if we take a look at trying to map this entire scenario to fire, Core, if you can advance real quick, there's still an awful lot of information. But if we pare it down and say, let's take one class, and we highlight, the next slide will highlight study design with a little blue mark. We can now start to say, okay, well, that's a small enough block of information to try and map. And if we look down at the bottom left-hand corner of the list of resources in Fire, we can see there's something called research study. So we open up research study and see if there's an easy mapping there. And it's close, but what we're really looking for is design criteria, not the overall study. And if you look at the third attribute down on the list, it says protocol, and it actually references another resource in Fire called the plan definition. So what we did is we opened up plan definition, and now we see that there is information within the plan definition resource that we can actually use to map back to our study design. And the nice thing about plan definition is that a plan can have multiple plans underneath it. So the idea of an overall study with multiple bits of information underneath it or multiple procedures underneath it kind of starts to fit in really nicely. So now we can go back and map the study definition or the study design class to the plan definition resource. And the first three items there are very straightforward. We don't see one for labels per se, and we don't see one for rationale per se, but there are values within the resource, the plan definition resource, that are appropriate enough. So we can label, we can map the label to, I think we used either subtitle or title, and rationale we mapped to purpose. So you can take a quick look at the next slide, and they're sort of delineated in red arrows to show that there's a mapping, but it's not exactly the same words, but it is a mapping that we can use. I'm going to take a look now. Let's bring in the idea of a population, or bring in the idea of a population. So once again, we look at study design, and the first place we go is resource study. We know that study design is somehow linked to the population definition class. And if we look closely, we'll see down at the bottom, there's something called enrollment, which sort of sounds a little bit like a population. And sure enough, we have a link to the group resource in Fire. And so now we can start to look at mapping population definition class to the resource group. So let's take a quick scroll down to that. And, again, there's information that maps nice and tidy over to what's in group, I want to drill down specifically on the last item in the population definition class, or at least the one that's shown here, and how that's mapped. If you could, yeah, you can see that I've kind of highlighted that as something to be further discussed, and it brings up the idea that in fire, something called bindings. And basically what a binding is, it's a way to take a resource element and map that to a value set or a code system or a code list, or if you're an old guy like me, you say like a pop-up list. But anyway, it's a list of valid values. So in the group resource, there's actually a couple of different terminology, a couple of different bindings that we can use. And one of them you can see is the group characteristic code. And that can be used generally across many different valid value or sets of valid values: gender, age, owner, location. I'm not going to use the one that says the yes or no, which is in the example that we have, but just to kind of extend the idea of what a binding can give you, I'm going to pick the planned sex attribute. So if we take a look at from the implementation guide, planned sex, and it maps out to the code list in SDTM, and there's a value for that, ends in 732. And if we take a look at that code list, sure enough, there are the 3 values for gender, male, female, and it's either both or undecided. But the idea that you can have multiple values tied directly back into the group resource. So we can take a advance to the next slide real quickly. I'm just checking the clock. We're coming down to the last minute. The last thing I want to talk about is the idea that we've sort of combined a couple of different classes, and that's because there's no exact mapping in Fire to map to every single class in USDM. What we've had to do is sort of work together and join multiple resources together. In the Fire implementation guide, or in Fire itself, it allows for this activity, and the concept is called an artifact. So if you take a look at the next slide, Core, what an artifact allows us to do is basically create complex relationships between multiple resources where those don't already exist. Fire is flexible enough to do that. Essentially what we're doing is creating custom artifacts, and in fact that lays the foundation for what we're really working on, which is an implementation guide for USDM to Fire. And with that, I will turn it back over, I guess, to Belinda, because we're at the end of our presentation with 7 seconds to spare.
Excellent. Thank you so much, Sam, Core, and Brittany, for a great presentation. I know at Transcelerate we talk a lot about bridging the gap between clinical research and clinical care, and I think what you just walked us through is an excellent example of how organizations are able to use USDM to support that bridging of that gap. So on that note, we'd like to turn it over to our second set of presenters today, which is Berber from ClinLine and Ali from EQTY Life Sciences. And just as a quick reminder to folks, especially anyone who joined late, you can submit questions at any time using the question function. And as soon as Ali and Berber are finished presenting, we will get to some of the questions that have been submitted. So please keep the questions coming. And on that note, I'll turn it over to Berber and Ali.
Thank you. Thank you for the opportunity to present our use case today. So my name is Berber Snoeyer. I am representing ClinLine, which is a consultancy company dedicated to the optimization of clinical research data processes. And my co-presenter is Ali Doetsen from EQTY Life Sciences.
Yeah, thanks, Berber. I'm Ali, like you said, at EQTY Life Sciences. We provide novel technologies like cryptographic governance, lineage, and traceability to any AI models or any parts of the clinical data flow. So back to you, Barbara.
Yes. So this use case that we are presenting today is based on a proof of concept that we did earlier this year, and based on the requests from Transcelerate, providing that with USDM, we further automated the process by using the USDM as the driver of this process. So we will start with an introduction, and then we will cover what content of the USDM was considered and are used for this use case. Then we will go step by step through the process from file source to SDTM submission datasets. And finally, we will talk about the corresponding lineage and governance processes. And then we will finish with our conclusions. So what are real-world data? Real-world data are, according to the FDDA, data relating to patient health status or the delivery of healthcare routinely collected from a variety of sources. And then the corresponding real-world evidence is the clinical evidence regarding the usage and potential benefits or risk of a medical product derived from analysis of real-world data. So we are also very active in the FUSE Real-World Evidence Working Group, of which I am one of the chairs. And in February, this poster was presented by the Quality and Reusability Project of the working group at the FUSE US Connect. And it summarizes very nicely the requirements and issues with using real-world data for regulatory purposes. So 2 months ago in July, the FDA guideline on using real-world data from EHR and claims data has become final, which makes it even more apparent that we should evaluate and use real-world data based on reliability requirements like, as you see here, traceability, completeness, and accuracy. Next slide, please. So in our proof of concepts, we have mimicked the data flow from EHR source to submission datasets by starting with the LZZT protocol and using the FHIR synthetic JSON files provided by Cynthia. Then we transformed it to OMOP-like datasets. OMOP is a standard which is often used for observational data, or real-world data is stored in a comparable structure in the data vendor's database. OMOP has some additional advantages that I will come back to later. After the transformation to OMOP, the data was anonymized and the patient cohort was selected based on the eligibility criteria defined in the protocol and stored in the USDM. Finally, those selected patients were assessed for fit for purpose, and the data of patients selected based on this was transformed to SDTM. Next slide. As you can see, for this process from source to SDTM, the standardization was very important, as well as the anonymization, adequate and transparent fit for purpose assessments, and showing the full lineage, traceability, and governance from source to submission. And Ali will dive a bit deeper into that last point in the next slide.
Yeah, thanks, Barbara. So in order to provide the reliability and traceability that we want over the whole process that we're about to describe in the next 25 minutes, we needed to find a way to prove that. So we found that cryptographic integrity is foundational to the interoperability and transparency in any process, and that's no different here. So how do we get data that is anonymized and from an EHR and through a data vendor to the sponsor into a submission, which then fulfills the needs of the regulators who want to verify the provenance of such real-world data, but without actually having access to see the data as it was at the data vendor. So I want to try and explain to you a little bit about how we're using cryptographic or digital signatures in order to do that and improve that, give that reliability and the traceability of the data. So as we look through here on the diagram, we have the data from the EHR going to the data vendor, which is then anonymized. We create a digital signature based on that, and we put that into our integrity fabric. That integrity fabric I will be showing a little bit later on, but it's based on content of the file. So if we take a data set, we create a digital signature from that, it's based on the content of that file. Now, if we've given that to the sponsors, they have the same digital signature, but if they change even one small piece of that data, that digital signature will no longer be verified and match at that point. So we have mathematical proof that the data that the sponsor is using matches what the vendor gave to them earlier in the process. So that's really how our digital signatures are working. We're getting mathematical proof based on the content of those files, whether that's code or data or any artifact, and we can see those in our complete lineage and traceability as we go through. So as part of this, we were using, as Berber mentioned, the LZZT protocol. I'm going to hand that back over to her to talk more about the USDM content.
Yes. So before we start with going through the whole process step by step, I wanted to indicate what information stored in the USDM was useful in our process. First of all, the eligibility criteria with tags to USDM attributes like a history timeline, biomedical concepts, population characteristics, and indications. And I also added tags to potential external coded dictionaries like for contraindications and contramedications. So in addition to that, the timelines and CDISC biomedical concepts were also very valuable in data selection for the patients. And then combining all that with the study design elements, it can drive the creation of the final SDTM datasets. In this use case, we focused on the eligibility criteria, but we also will indicate where USDM is useful in later steps. So here you see for the inclusion criteria, if you can go through to the next slide, what information I tagged from the eligibility criteria. So in the different colors, you see the population characteristics. In green, in red, you see more the biomedical concepts, and also a light green timeline tags. But of course, they had tag names, which is presented in the next slide. Yes, so you see that I replaced that with tags and tag names. The corresponding tag values were stored in the background in the USDM. Next slide. So, but before we could do the eligibility evaluation, we needed to select the JSON files of patients that had the right condition. In our case, this was Alzheimer's disease. And here you already see that we needed to specify more than one code in USDM to select the complete group of Alzheimer's patients. In USDM, you can store as many codes as you need for that. So with the codes linked to the indications stored in USDM, we have automatically selected the patients from the source. Of course, if we would change that indication with other codes, then we will get a completely different set of patients from the source. For governance, we also outputted the selected list of patients, which could then be verified by the data owner. Next slide. So then step 2 is needed because we needed to standardize the JSON source information files per patient, which were per patient, into structures that can be used for transformation and evaluation. In our case, we used OMOP as a basis for that transform datasets because this already includes a lot of source information variables. And also formats that are used at vendor sites often have the same kind of structure as the OMOP database, so that also fits in our use case. We added more variables from the source when needed for traceability and left out complex OMOP datasets and variables that were not needed. So hence, that's why we call it OMOP-like. We used standardized mapping input and algorithms to transform the data automatically from FHIR to this OMOP-like structure. Next to you.
Yep, so how do we start to create the reliability and the transparency of what's happening in this data flow? So on one side, we start to take the FHIR, the JSON files for each patient. We start to feed in the JSON files. We can read that using some kind of code. And we start to represent this and build this up on our Lineage Explorer and the Integrity Fabric. So this is kind of what it starts to look like, and we'll delve into this a little bit deeper later. So we see what it looks like as we just take individual patient files. We're taking the output of what's in the JSON file for the FHIR format from the USDM protocol, and we start to build the OMOP-like database. So as we move through, we'll see more of these snippets as to how this progresses through the lineage flow and to create that traceability. So the next step that we start to look at is around the anonymization. So we have the codes, we have the patient data. How do we start to give that a little bit of privacy around that? So again, we're using cryptography to do that. We're building code. We're using SHA 256 algorithms to create that. So according to the rules that we wanted to set up for this, again, we have governance set around these rules. We wanted to scramble the ID variables. We wanted to shift the date and time variables. And we wanted to remove any potential sensitive information. We did that using a script. We did it on a trusted execution environment. We get audit and verification of the privacy and the integrity of that data coming through. So the output on the other side was a similar database, but now anonymized. And we started to use that. It's linked to FHIR, and we then use that throughout the rest of the process. Okay, Berber.
So yes, so step 4 is the most important one in this use case regarding USDM. So here we show the automated selection of eligible patients. Before I showed you the mapping of the eligibility criteria, and here you see how that is stored in USDM. Those tags that I showed are linked to the corresponding classes and attributes via the specifications in the parameter map class. So in our case, it points to study design population, which you also saw in the previous presentation, timeline and biomedical concepts, but we also had some tags that were only a specification of text, contraindications and contraindications. So based on these specifications, we could then subselect the eligible patient cohort and define the corresponding index date. Next. So based on the naming of the tags, I have linked them together. So here you see the biomedical concepts that I tagged together with the allowed minimum and maximum values and the corresponding units, which were also stored in that parameter map class. The units were also in an issue because the unit in USDM can of course be different from that one, from the unit that is used in the real world data source. Next slide. So those biomedical concepts that I tagged were stored in the USDM together with their standard CDIS codes and corresponding alias codes. Those alias codes can be used for selection of data from the source. And here you see the alias codes that were stored in the USDM for a number of the BCs that we mapped. In addition, because we used the OMOP format, we also needed to transform those alias codes automatically to OMOP codes using the OMOP concepts dataset, which is available from the Athena dictionary. Next slide. The automated selection of BCs from the source can be performed as long as you make sure that the codes used in the source are defined as aliases in the biomedical concepts stored in USDM. It might be cumbersome to specify all the potential codes for a BC that could be used in real world, but in this regard, the Cynthia data was very ideal in comparison to real world data use cases that I've been working with in the past. However, one of the nice features of OMOP is that you also can use OMOP to select all the children of a BC code. So then you don't have to specify all the different variants. So then only the parent has to be specified. This presentation is too short to explain, but feel free to ask me later. So in our case, we used SAS to do this automated selection, but of course, this can be done in any tool or any language. We automatically selected the available measurements, did a last value carried forward, used our pre-specified min and max values and checked the units and performed the corresponding unit conversion if needed. And here you can see an example of the data set created for this. I hope it's not too small. But this female patient apparently got an Alzheimer diagnosis when she was 49, which was not within the eligibility criteria for age, which started at 50. Then the MMSE value, which is BC1 in this case, was also still too high. So she was still too good. She was not ill enough. And BC4, which is SDOT in this case, was also not within the limits at that time. However, when she turned 50, the SDOT was measured again and within the limits, and also the MMSE score, which was measured a bit later, had dropped. So it was within the limits as well. At that moment, the patient was eligible for the study as far as the available information concerned, and the index date could be set to the date of the MMSE measurement when all criteria were met. So the reasons that BC3 and BC5 are empty in this case is that these were evaluating the male-specific criteria, so they were not applicable for her. However, if a measurement was not done, then we also did not exclude the patient in our use case. And of course, decisions upon this and allowable timing windows of measurements can be set and are at the discretion of the study managers. Next slide. Here you see the output of this step with the corresponding attrition diagram, which is a required aspect of study designs in real-world studies. So you see that we started with 159 patients, and then when we did all the selections in our use case, we ended up with 85 patients. But what happens if we change one of the eligibility criteria? We automatically do that in USDM, export USDM as an API, and then the number of patients changes. And then you see, of course, in this case, we have a lot less patients, only 43 left at the end. Next slide for
Yeah, thanks, Berber. As we continue to go through, how does this look? How are we starting to subset down as we move through the process? Again, we can see the eligibility criteria and biomedical concept references. They flow through from earlier on in the lineage flow. The anonymized OMOP-like database is used, and we can use the cohort selection program to really subset down what we're looking at. But the outputs on the other side, we see not only the anonymized eligible patient cohort, but also we get the attrition summary and we get the patient selection report that we can see and we can link to wherever they're stored. Again, storage doesn't really matter. This is held in this integrity fabric. Okay, it's the windowing.
So after the cohort selection, we did the fit for purpose assessments. And for this, we aligned the timings of the real-world control arm with the interventional arm that was defined in the Lilly protocol and also stored in the USDM. However, we limited the gaps. We changed the gaps between the timing to be sure that we had as much data as possible from the source. And we created rules in case more real-world visits were available in a window. Take the one that is most closest to the middle. And we defined the minimum period of data required as well as the minimum number of visits with data required. In addition, you can have more fit for purpose assessments, which may include data validation, bias assessments, missing imputations, and matching of real-world control patients to the interventional arm patients. Next slide. So then when a subset of patients and data is selected for the final evaluation, this subset can be mapped to SDTM. Limiting the number of patients and variables to what is needed for submission makes the SDTM mapping less burdensome and limits the effort needed for this. So we used the same kind of mapping process as presented for the OMOP mapping, but as an alternative, we didn't have time for that. The specifications stored in USDM, like trial design, information timelines, biomedical concept information, can be used to cover a big part of the mapping. Especially the CDISC biomedical concepts include already information on the final SDTM dataset that it goes to, variable and units and also the variable and the units in which the SDTM data is expected to be stored. So that's very helpful. Next. So our last step in the proof of concept was to show the auditable traceability of the data. To show this, we mapped the information stored in the SDTM vital signs dataset back to the source via the OMOP-like datasets. I'll let Ali talk about the de-anonymization, but before I do that, I like to show you the resulting FHIR JSON code matching the original files, which is displayed on the right of the screen. So all the black information that you see there was reproduced reproduced back from the end. Only the red information, not as it was not deemed necessary at the beginning or proportional at the beginning to use it, and therefore it was not included in the original mappings. Over to you.
Yeah, so because we're using those cryptographic or digital signatures in order to create the anonymized data and to take that forward, we have the ability to, again, match on those codes going backwards. So we always have that mathematical proof that what happened was actually what happened, and we'll always have that proof. So as we start to do the reverse mappings, that's where that kind of comes in. It's really useful to go back to the beginning using that cryptographic methods. Okay, so for the last part of this presentation, we're just going to flip over and do a quick demonstration just to show you how we've actually traced the flow from beginning to end. So this is where it always becomes a little tricky as we flip from one to the next. Okay. So this is what the Lineage Explorer looks like. It's very zoomed out at the moment just to give you a flavor of what this looks like from end to end. The very left-hand side, we have the USDM protocol, an associated program to read that in. And on the right-hand side, we have the SDTM mappings. And we're just going to step through and take a look throughout the process as to what's happened. We'll also look at the governance processes that were assigned to it along the way. So we can see now as we zoom into this Lineage Explorer, we can see we've got the USDM protocol on the far left. This is the JSON file. We have a computation node that then takes that and splits that, and we can see the result of those being multiple data sets, including the dictionaries, activities, biomedical concepts, and so on and so forth. Because this doesn't really matter where the data is stored itself, this is almost metadata, if you will, about that. The digital signatures representing that data itself. So this comes from multiple different sources that you can see, and as we process these along. So we start to drill in, we take our individual patient data, we make a computation on that using a particular SAS program in this case. Other programs are available to do such. And then we started to get our OMOP-like database coming out of that. Again, we get our population overview as an output, and we start to build up our different data sets. So if I just go into and take a look at the death data set, we can start to see that we can see the person IDs, we can see the death dates as we go through that. That's not what we're going to see later on. We move through, we get to the anonymization step. So there's an anonymization Python code here. If I go in and I take a look at the computation node itself, kind of doesn't really make much sense. But when I start to drill into it, what is recorded on a ledger? What is the information that's being stored immutably so that it can never be changed? We can see here we have a computation, and we know that this has 9 inputs. So it has 9 different files that are inputting into this computation, and we can see then it has those 8 outputs that come afterwards. So those are the OMOP data sets. The SAS code is included, any other code and data sets are all included in these. And these are time-stamped, the credentials, everything is in there. I don't expect you to really take too much notice of that. But again, you can see here from this diagram really what's feeding in and out of that. And then once we get to this, we can see there's an anonymized death data set. So if I click into that, we can now see that the person IDs are scrambled, as we suggested earlier, and the death dates have shifted, in this case by a couple of years as specified in the program. So this can go through, the same applies to all of the other OMOP-like data sets. At this point, we can see there's the cohort selection data set. So this is using those USDM inputs to create that subset in the cohort. And the result of that is we get the subset of data sets, but we also get the summary of attrition values for the cohort and an output of the patients, as we suggested that we would get throughout this journey. We then take the windowing. So again, we're looking at fit for purpose. It boils that down, and we start to then take those data sets and build the SDTM. So the last few steps here are really just around building that SDTM. Now, how did we govern? How did we make sure that we were doing the right thing along the way? And how do we apply governance to any real, digital SOP, if you will? So if I come into the compliance section of the dashboard here, we can see there's a real world evidence reliability one. I can see here there's different sections that we went through. So licensing, transformation and mapping policy 1, anonymization, data cohort selection, and what really was done to govern those. You can see down here there's a bunch of these not started or in progress, but we've got one here just as an example. Ensure the detailed cohort selection is in the contract. So this is the very first part. It's the first one of 10 that we've got in this group that we want to do to ensure that what is happening is supposed to be happening, is governed by an SOP or governed by a policy. We want to control that. We can reference that document, so we could reference the contract. I've been in here and I've said, okay, the detailed cohort selection is included in the contract. I've made an attestation that has happened. Somebody's gone in and made a review. Yeah, it looks good in the contract, and we sign that as an outcome. And what that does is this is giving us this, again, attestations given in this governance studio to show that we've followed the process, and we can easily see that we've followed all of the compliance steps to make sure that we've followed the right SOPs to make this happen. Okay, I'll flip back to the presentation. Okay, so in conclusion, I think what we've shown there today is that the USDM and cryptography together enable a trusted DDF solution. We can get the automated patient selection from source. We start to automate eligibility and feasibility checks. We're automating data selection using the windowing, the biomedical concepts, and we can do SDTM mapping. We didn't show it in this particular proof of concept due to time, but we can also start to automate some of that based on that USDM. We're starting to get automated documentation, and we're getting verifiability that what actually happened is what we said was going to happen, documented by SOPs. We're getting that automation through using that digital protocol. We're taking that JSON file, and that's starting to drive our programs, drive the data flow throughout. That's giving us faster study design. It's giving us maybe better SDTM mapping, more automation happening throughout that. We're getting more accuracy, getting better accuracy in what we're trying to do, but we're also getting this cryptographical automated lineage. We're getting process traceability, and we're getting those governance controls around the topic that we're going through, whether this is using real world and getting the reliability or any other data flow or AI models. So the last thing really to say is that USDM can inform real world data, fit for purpose assessments, and that will ensure suitability for real world evidence so that this can be used in submissions rather than just supporting submissions. Okay, thanks everybody for your time today. I'll hand back to Belinda.
Thank you, Ollie and Berber. So as we mentioned, we'd now like to open it up to questions from the audience. I know we've got quite a few that have come in through the chat, so I will, or through the question function, so I'll get started with a few that have already come in, but I would encourage folks to continue submitting questions if you have them for either for NNIT or for EQTY and ClinLine. So based on the questions that have come in already, I'm going to start with one that I think actually is for both sets of presenters that came in. And the question was, currently protocols are written in natural language. How will the data in the protocol get into a digital machine readable format? For example, into a format that something FHIR can use. And again, would be interested to hear perspectives from both sets of presenters on that topic.
Yeah, I can give our answer first. And so again, this is pioneering work, meaning that there are actually multiple ways of achieving that. One way could be through the utilization of structured templates for manual or automated data entry. Could also apply NLP tools to extract some of this data from the text and then convert it into FHIR resources, etc. Or we could even utilize tools or platforms supporting structured authoring of protocols from the start, and then make them compatible with FHIR.
Thanks, Cor. Would maybe Berber or Ollie, any perspective on that one?
Yeah, I think we more and more see also study design tools that also create the document on top of it. But yeah, I think I also like the more the template like approach. But yeah, we see more and more that people are interested in that and transitioning to that. So, but also a lot of historical protocols are being parameterized so that you can use that as an library to set up new trials.
Thank you. So I think that leads into another question that came in specifically for NNIT that's sort of related to this. It says, does the use of HL7 FHIR assume the use of Transcelerate templates for protocol and SAP.
I can take that one. So we did the mapping that we presented based on the Translator e-templates. So our assumption is that that should be the future, and that should be the approach. But much of the information that's captured in protocols is kind of similar to the e. So you could map it to FHIR even if you're not using those templates. But I think that's the recommended approach.
Great, thank you for that.
Just to expand on that, if I may. So the short answer is no, like Brittany said, does not require it. We need to keep in mind that FHIR is a data exchange standard. That's a very important point. So the two can indeed work together if need be, but FHIR can also be implemented independently of e-templates.
Excellent. Also, thank you. So I think, on the topic of sort of what's required to enable the two solutions that were shown today, switching back to Berber and Ali, I think really great walkthrough on sort of how USDM can be used as a starting point to enable RWE and RWD. You talked a little bit about sort of the role that the BCs that are embedded in the USDM play in this. So how critical are biomedical concepts to the approach that you showcased? And so specifically, would it be possible to do what you showcased without the use of biomedical concepts?
That would be very difficult, especially because you need all those codes from all those external dictionaries. So it's biomedical concept really represents all aspects of an assessment that you need to get the right information from the source. So if it wouldn't be stored in the USDM, I would store it next to the USDM, but I think how it is stored now in the USDM with the corresponding alias codes is very beneficial to this process. So could be done without that, but then we need to add something next to it, and yeah, it works very well now.
Excellent, thank you for that. Switching back to NNIT, then we had a question that came in. I think you had made a reference to C-dash. So we had a question that came in that said there was guidance released on C-dash to FHIR mapping, but how are C-dash and USDM related, if at all?
So the USDM is the standard that can be used to digitize the protocol information, kind of that higher level, like the study design. And then C-dash can be used to structure the CRFs, the data collection forms, based on that protocol. So they're similar in that they're standards, but they're complementary. So using C-dash and mapping it to FHIR can ensure that the data collected during the trial is consistent with that digitized study design defined in USDM.
If I may add to that, so both are of course CDISC standards, so that makes it more easy to convert one standard to the other and to translate the information from USDM to CDASH.
Excellent, thank you. And then I think another question that came in for NNIT was around how will you use protocols in the EHR? Will you create an EDC like? Will you create EDC like fields in the EHR?
Yeah, that's a very good question. So I guess the short answer is that protocol data will be represented in the EHR using the FHIR resources Sam mentioned. So it could be the plan definition, etc. And that will, of course, allow tracking of patient care pathways and study-specific activities. But it is very important to mention that the aim is not to replicate all EDC fields, but to only include the relevant protocol information that is important for the patient care itself. So the data elements that are shared between the EDC and EHR will be mapped appropriately, but the EHR will only include clinically relevant information. So again, back to the general and soldier's analogy.
Thank you. Sorry, we've just got a few questions coming in that I'm just trying to parse through, so just bear with me for a second. Here's a good general one, I think, for both presenting entities today. For companies that have not yet undertaken any DDF initiatives, where would you recommend they start?
That's, I think, one of the most difficult questions. So you need to become familiar with the data. So I think starting with what information you need for your downstream processes, where is your first need to optimize, and then look if you can digitize that information, or with the protocol kind of template, or with a study design, or extracting that from a protocol or something like that. But yeah, enabling that storage in a USDM format, and then setting up your solution, and then you can build up on that. But yeah, find the most critical use case first.
Yep, totally agree. Start with data and data flow. That is, I mean, regardless of what application you're using, but in this particular case, because you're at the end of the day, we're talking about moving data around. That's exactly where I would suggest starting as well.
Right. Thanks, panel. And then another one that has just come in, and maybe we could start with Barbara on this, but again, be interested if NNIT has a perspective on this as well. Question is it possible to have a USDM catalog that would be used for several studies of the same type, or should a USDM be defined for each study?
That's a good question. I think you can set up a catalog which is structured like USDM, but then USDM also is an exchange kind of format. So you have the data model and you have the API. So you can then store the specific study in one version and exchange that. But the catalog indeed can be set up to reuse this information for different studies, multiple studies.
Any thoughts on that one from NNIT?
Ditto.
Sure. Oh, Sam, go ahead. Oh, you said no? Okay. Well, yes, it's possible to have a catalog, and like we just heard, it should act as a template or reusable model, but of course only for the more common elements. So it could be stuff like data definitions, structure, protocol components, etc., that are more or less consistent across similar types of studies, maybe disease area, study designs, etc. So short answer is yes, it is possible.
Thank you. And then I think maybe just one last question for today, and then we do have a few closing announcements. This one actually came in specifically for Berber, but I would again open it up to the full panel for anyone else that would like to weigh in. Have you had any discussion about a blockchain solution for the digital data flow with wallets?
Maybe more for Ellie then.
Yeah, I think so. So there are subtle differences between the common perception of blockchain and the baggage that comes with that. I think we prefer to use the terminology of applied cryptography. It's a little bit simpler and easier to work with. So no, doing a direct blockchain with wallets is not something that we've discussed at this point. We're using the cryptography and the digital signatures to get the compliance and the lineage flow through it, not using blockchain to do that.
So maybe to add to that, one of the most important parts of using clinical data, you're really using real world data, is that you can trace it back to the source. And blockchain has some more problems with that, while cryptography has that option, as a better option for that.
And just a more general answer, this industry that we're in is often really slow to adapt to big leaps in technology, vis-a-vis the fact that we're still working with PDFs as our primary means of moving data around. So um, uh, technologically speaking, I totally agree that that's a path, but from an option perspective, I would think it would be really difficult for particularly larger companies to want to dive into something like that without a lot of time and energy into how it would work.
Thanks, Sam. I think some great perspectives on that one. I love that really today we've really touched on a lot of hot topics, everything from RWD, RWE, bridging clinical research and clinical care, and closing it out with blockchain. And I think there were a few references to AI in the process as well. So certainly, USDM is front and center in terms of enabling some of these really exciting trends in our industry. Thank you then to all our panelists. I would like to just make a couple of announcements before we wrap up today, specifically just to make everyone aware of a few other upcoming events that we have. So Lauren, if we could go to the next slide. And if we could go one more, thank you. So as I did mention in the intro to this webinar today, this webinar is actually the first in what we intend to be a series of DDF Solution Showcase webinars. With that in mind, I would like to announce that we've already scheduled the next of these webinars. It will be held on December 5th at 9 a.m. Eastern Time. We will be announcing the two presenting organizations who will be featured in that showcase shortly, so please stay posted for more information and details on that. And obviously we'd like to invite everyone to attend the next one in this series. I'd also, if we could go to the next slide, just like to make everyone aware that Transilio will also be hosting a webinar on October I believe it's the 15th. Yep. This will be featuring the Vulcan Utilizing the Digital Protocol initiative. And specifically spotlighting some of the findings from the recent Connectathon that was held earlier this month. So for this group, anyone interested in that topic that obviously relates very closely to what we're doing in DDF, would certainly invite and encourage folks to attend that webinar as well. And if we could just go to the next slide. And finally, just wanted to make everyone aware of the various tools and resources that we have relating to DDF. So hopefully for maybe some of you that are on this call that are relatively new to the Digital Data Flow Initiative, this may have piqued your interest to learn more, and we certainly have plenty of resources available that you can reference to learn more about the initiative, various ways to stay connected as well. Dedicated GitHub page, feed on social media, etc. So again, would definitely encourage folks to take advantage of some of the resources that you see listed here. And finally, if we could just go to the last slide. I just want to extend a big thank you, both to our audience that participated today. Looking from the numbers in the GoToMeeting that we had a really great turnout today. But more importantly, I'd like to extend a huge thank you to all our participants, presenters, panelists today. I think really some very informative and thought-provoking content that was shared today. So just want to again extend a huge thank you to everybody that presented and participated in the panel today. Thank you all so much, and thank you to everyone for joining.